Hi class, so today's video is on protein structure, which we've been covering in class. In this section you have to know the 4 levels of protein structure and their significance. You have to know the difference between fibrous and globular proteins. You have to know the difference between polar and nonpolar amino acids and their significance. And you have to know 4 functions of proteins. And the examples of the 4 functions are given on page 207 in the table, so you should know 4 general functions, and so it gives some examples: hemoglobin, actin and myosin, insulin, immunoglobulins, and amylase. So you should make flashcards. So you should know the 4 functions and then give an example of each. And as I said, that's given on the table on the top of page 207. So to start off, you need to know what hydrophilic means and what hydrophobic means. So hydro refers to water, and philic refers to liking. So molecules that are hydrophilic like water, and molecules that are hydrophobic, phobic refers to fear, right, don't like water. So in an amino acid, the R side chain, so if we draw an amino acid, which you should know how to do from memory now. So that's the carboxylic acid group, the R side chain, and the amine group. The R group changes and then has different chemical properties, and the different chemical properties that are in the R side chain can change the structure of the protein. So some of the R side chains are hydrophilic, and some of them are hydrophilic. If they're hydrophilic, some of the R side chains are hydrophilic, and some of them are hydrophobic. If they're hydrophilic, they undergo certain kind of bonding, and if they're hydrophobic, they go through another type of bonding, and we will talk about that when I talk about the polar and nonpolar amino acids. So to go through the structure here, the primary structure is the sequence of amino acids. So, and the sequence of amino acids is determined by the DNA sequence, and we haven't talked about how we go from DNA to protein yet, but we will. But for now, all you have to know is the primary structure is determined by the sequence of amino acids, and this picture is small, but you can see here that there's an R1 and an R2 and an R3, and that is what we've been doing in class. We've been building these long chains. The secondary structure are interactions of the backbone, the long chain part of the protein, not the R groups. So the R groups are not interacting in the secondary structure, but it's the side chains. And you see the part that I'm circling here, those are the hydrogen bonds between the carboxyl group and the amine group that are found in the backbone of the polypeptide. And there's 2 types of secondary structure that are most common. They are alpha helices, so it looks like a helix, right? It looks twisted up upon itself. And you can see an example of a 3-dimensional helices when in the classroom. If you look up to the ceiling, you see that there's DNA double helices hanging from the lights, and it's somewhat similar to that. For beta sheets, they interact more of like along long chains, and so you have kind of this flatter structure, and they call them beta pleated sheets because they bend up and down kind of in like a zigzag pattern, but generally flat. In the tertiary structure, you have interactions between the R side chains. So there's interactions between the R groups. And on page 208 at the bottom, it talks about the different kinds of bonds that can form between the R groups. So one of them is covalent bonds. When there are sulfur atoms, cysteine is one example of an R group, and it has sulfur in it. And if you have cysteines in different parts of the structure, they can actually bond together and form what's called a disulfide bond. So I will write the list of possible interactions that happen in the tertiary structure. So we have covalent bonds between sulfur atoms, which are in the cysteine amino acid, and they are called disulfide bonds. It's just the name for the covalent bonds that form between cysteine amino acids, or S atoms. And these are pretty strong, as you know from chemistry class. You can form hydrogen bonds between hydrogens of one part of the molecule and oxygens or nitrogens in another part of the molecule. And these are relatively weak, but if you get a whole bunch of them, it can be quite strong. You can have van der Waals interactions, and these are actually happening between hydrophobic side chains, hydrophobic R groups. And that's what it says there on page 208. So usually you'll find these pointed inwards in the cell when there's water. The hydrophobic side groups will be pointed away because they don't like water, right? They're hydrophobic. And you can also have ionic interactions. So if you have a positive charge side group on one part of the molecule and a negative on the other side, you can have an ionic bond that form between positive and negative charged R groups. So the big takeaway message is that there's many kinds of interactions that can happen to form the tertiary structure, but the most important thing you have to know is that this happens between the R side chains. So, and then the last level of structure, if we go back one slide, you'll see over here in the quaternary structure, they have different color proteins, and actually these are different polypeptides, and they're interacting with each other. Now, in this example that is used here, they're using an example of hemoglobin. So you see heme, which helps bind the oxygen. There are 4 different proteins that are interacting together. So they're acting like in cooperation or in concert together to help bind oxygen in the blood. So the next objective that I'm going to discuss is, what is a fibrous protein? What is a globular protein? So it's kind of in the name. Fibrous means it looks like a fiber. There are long chains of proteins, and they are insoluble in water. And the book gives several examples. One of them is actin, which is involved in muscle contraction. It's also found inside lots of cells. And another example is collagen, which plays a structural role in connective tissue. So, for example, you're all very young, so you have lots of collagen in your face to make sure you don't have wrinkles. But as you get older, the collagen breaks down, and so the skin isn't kept as tight. That's because there's less collagen holding the skin tight. So for globular proteins, you can think of the word glob, and they are more three-dimensional in structure. And obviously, I can't draw 3 dimensions on the screen, and these are soluble in water. And the examples they give here are insulin, which is a hormone which signals the body to break down sugar. And the other example they give is hemoglobin, and in England they spell it the way that the book writes it, but in the United States we write hemoglobin this way. Either way is fine. But hemoglobin helps bind oxygen in the blood. So the significance of this will be more obvious as we go on. But what causes these structures? Why are some long threads fibrous and some form globs? Well, it has to do with the structure, with the characteristic of the R side chain. Are they polar or are they nonpolar? So, of the 20 amino acids that are commonly found in humans, and in many organisms, of the 20 amino acids that are commonly found in all organisms, some of them have polar characteristics. That means that when you divide them, that they're asymmetrical. Some of them are nonpolar, so they are symmetrical in nature, so that they have. So nonpolar molecules tend to be hydrophobic. They like to stay away from water. And polar molecules are hydrophilic. So think of water, right? Water is asymmetrical. It has a negative side and a positive side. So it's going to be attracted to water. It's going to be attracted to other water molecules. But a molecule like carbon dioxide, which I'll draw here, when you split it down the middle, it's even on both sides, so it is not going to be attracted to water. It's going to stay away from water. So in the case of when you put these R side chains together, some of them will like to interact with each other. The ones that are hydrophilic will like to interact with each other, and they form hydrogen bonds and ionic bonds. And the hydrophobic ones will want to stay away from the polar ones, and they will form van der Waals forces, or you may know them as London dispersion forces. And the idea is that the polar ones stick together and the nonpolar ones stick together, and that in turn forms a three-dimensional structure. And that'll be obvious when we do the pipe cleaner activity on Thursday and Friday. So I hope that was helpful in decoding what was in the book. Please write down 2 questions that you may have, and you will turn that in to prove that you watched the video, and we can have a class discussion about the video. Thank you. Thanks. I'll see you soon.